right, I just wanted to get into this 2015 paper that we had, mainly led by Zinlin Yu, and it's an amazing study that's very mechanistic about how the proteasome actually assembles. The key takeaway is that a lot of the assembly of the 19S within the lid is really what drives correct assembly of the proteasome. And with that, let's dive into the Notebook LM summary of the paper.
All right, let's begin our technical audit of this 2012 paper by you and colleagues. The central claim here is a revised assembly pathway for the 19S regulatory particle lid. Our analysis is going to be strictly results-oriented, focusing on the methodology and the interpretation of their core data. So our framework is pretty straightforward. We'll start with the core question the paper's trying to answer. Then we'll critically examine the experimental systems they used. After that, we'll dive into their two main findings, this lidless intermediate and the role of the RPN11 C-terminus, before we assess the in vitro validation that they used to support their final model. This paper is really addressing a fundamental question in proteasome biogenesis. They're asking what's the precise mechanism for how module 1 of the lid nucleates, and what really governs its association with the base CP complex before module 2 even gets recruited. Okay. Any claims are only as good as the experimental design, right? So let's really dig into the engineered strains of Saccharomyces cerevisiae that they've leveraged to try and dissect this assembly process. So looking at this excerpt from their Table 1, their primary tools are pretty clear. They're using tetracycline-inducible knockdown strains for RPN8 and RPN11, which are core subunits of the lid's MPN domain. And critically, they're also using this RPN11-1 mutant, which, as you can see, expresses a version of RPN11 that's truncated at its C-terminus. Now, the gene silencing protocol itself, it's a standard and, frankly, very effective approach. You add tetracycline that represses the TET02 promoter, and that leads to a conditional and titratable depletion of your target protein. This is a great way to observe assembly defects before you get into issues with cell lethality. So, this setup brings us to their first major result. The authors used this knockdown system to probe the direct consequences of depleting these core lipid components, RPN8 and RPN11. Let's take a look at the native page from Figure 1A. This is where it gets interesting, because this separates protein complexes by size and shape. And what you see is a critical finding. When you knock down either RPN8 or RPN11, the bands for the fully assembled proteasomes, that's both the doubly capped RP2CP and the singly capped RP1CP, they're significantly reduced. And in their place, you get this new, faster migrating species that they've labeled base CP. So the immediate conclusion you can draw from this gel is pretty clear. Both RPN8 and RPN11 are absolutely indispensable for starting lid assembly. You take them away, the process stalls, and you get this accumulation of base core particle complexes that just can't form a complete lid. And this is where the study really shows its rigor. The identity of this new band isn't just an inference based on how it runs on a gel. The authors went ahead and confirmed its composition using mass spectrometry. And as they detail in Table 3 of the paper, it contains all the base subunits, but is completely devoid of lid components. Okay, the second key finding comes from their analysis of that RPN11M1 mutant. This experiment gives us a much more granular understanding of RPN11's function. It's not just about simple initiation. This in-gel activity assay from Figure 2b is quite telling. It lets you see proteolytic function right there in the gel. What you see is that the RPN11-M1 mutant assembles a very distinct proteasome species labeled alpha. Now, it's proteolytically active, but it migrates faster than wild type, which immediately suggests an incomplete complex. And sure enough, their follow-up MS-MS analysis confirmed this species is specifically missing the module 2 subunits, RPN3, RPN7, and RPN12. And then we have this rescue experiment in Figure 3b, which is, just a really elegant piece of work. They take cell extracts from the RPN11M1 mutant and simply incubate them with a purified recombinant polypeptide that is only that missing C-terminal fragment. And that alone is enough to restore the formation of fully assembled 26S holoenzymes. It proves that this fragment is what recruits those missing module 2 subunits. So what this all boils down to is a really clear delineation of a dual structural role for RPN11. Its MPN domain anchors it right into module 1, and that allows that module to dock onto the base CP. But the C-terminal helix acts as a physically distinct tether, and it's absolutely essential for recruiting and attaching module 2. All right, so to really nail this down and validate the assembly model rigorously, the next logical step is to move away from the complexity of cell extracts and into a fully defined recombinant in vitro system. And the data presented here in Figure 4 are pretty unambiguous. They demonstrate a strict, stepwise assembly pathway for Module 1. The whole process unequivocally kicks off with the formation of a stable RPN8, RPN11 heterodimer, and only after that do you get the sequential addition of RPN5 and then RPN9 and RPN6 to form the complete 5-subunit module.
Now, Figure 5a provides the critical link in the chain. The authors incubated various Module 1 subcomplexes with purified base CP, and the result is crystal clear. Only the complete 5-subunit Module 1 that contains RPN6 is competent to bind the base CP. This really singles out RPN6 as having a key role in docking the lid core onto the base.
All right, let's synthesize all of these findings into a coherent technical summary. This really does revise the existing model for how the proteasome lid gets built. So to recap the revised model, assembly is initiated by the RPN8, RPN11 dimer. Module 1 is then built sequentially onto this dimer. The entire preformed module then docks onto the base CBP, and that interaction is mediated by RPN6. And finally, the RPN11 C-terminus acts as that distinct tether to recruit Module 2 and complete the whole lid assembly. And this structural model in Figure 6 provides a really compelling visualization of how all these biochemical findings translate into a three-dimensional architecture. You can see RPN11, there in green, positioned centrally. Its globular MPN domain is coordinating Module 1, while that distinct C-terminal helix is pried away, perfectly positioned to wrap around the RP3, RP6 coiled coil and anchor Module 2. So, what's the broader technical significance of this revised assembly model? Why does this matter? For the ubiquitin field, the crucial contribution here is the identification and characterization of a stable, functional assembly intermediate, that Module 1 base CP complex. By dissecting the very distinct structural and functional domains of RPN11, this work provides new, specific targets for understanding proteasome dysfunction. It offers a much clearer, more detailed roadmap for studying the regulation of this essential molecular machine.